Hello, ladies and gentlemen. Today it is our pleasure to share with you our research proposal, which relates to the propagation of tau proteins in slow versus fast progressing Alzheimer's disease.
Throughout the experiment, our group will attempt to address the following question. Are there differences in tau propagation between samples of postmortem tissue from fast and slow progressing Alzheimer's patients? Before introducing our experiment and hypotheses, I would first like to talk about the impact of Alzheimer's disease. Alzheimer's is one of the most common causes of dementia that leads to impairment of behavioral and cognitive function, including language, memory, comprehension, attention, judgment, and reasoning. Currently, about 6.2 million people in the United States are affected by Alzheimer's, but it is projected that by 2060, this number can increase to 14 million. So one of the main pathological hallmarks of Alzheimer's disease is a protein called tau. When normal tau proteins are misfolded, they can block synaptic communications between neurons and eventually lead to neuronal death, which can affect several regions in the brain, including but not limited to the entorhinal cortex, hippocampus, and the cerebral cortex as the disease progresses. Moreover, Alzheimer's can be classified into two categories, fast and slow progressing Alzheimer's. Firstly, the impact on fast progressing Alzheimer's patients can be extremely grave due to the rapidly developing dementia, as most of the patients bear the responsibility of supporting their families and are living some of the most important parts of their life. Secondly, slow progressing Alzheimer's can not only create some uncertainty among patients and their families about the long-term impacts of the disease, but also affect clinical trials which aim to measure treatment effect within a fixed and short amount of time. When too many slow progressors are enrolled in the trials, they can sink a study, even if the drug did what it was intended to, since it would be really challenging to detect any response of the drug to abnormal tau activity. Therefore, by understanding the differences in tau propagation between fast and slow progressing Alzheimer's patients, we will be able to better understand the tau pathology in these two groups. Additionally, our potential findings can make a positive difference in the Alzheimer's community, along with possibly reducing setbacks in clinical trials.
Okay, now for what is known and what is unknown. So as Natrang touched upon, while researchers know Alzheimer's disease is not spread through contact with others, evidence exists that it certainly has an infectious component, somewhat related to the mechanism of prion propagation. Alzheimer's disease is well known to feature neurofibrillary tangles that are composed of misfolded tau protein. Analogous to the infectivity of prions, recent evidence suggests that misfolded tau spreads by cell propagation through the brain by means of oligomer seeds that travel across a synapse to another nerve cell. The total amount of abnormal tau in the AD brain is linked to disease stage and severity. A critical aforementioned hole in this hypothesis, however, of how the disease spreads is that it doesn't explain why Alzheimer's progresses at different speeds in different patients. And now to dive into some details of our experiment. So there are two central hypotheses that were created to explain this difference in Alzheimer's speed of progression. One hypothesis is that Alzheimer's spreads slower in some patients because they are genetically predisposed to have immune systems that are more efficient at disposing of misfolded tau and preventing its propagation within the brain. The other hypothesis is that the patients with slow progressing Alzheimer's simply develop less aggressive forms of misfolded tau, thus causing propagation to occur at a reduced rate. Because existing research suggests that there is great variability of tau oligomers that can form within the brain, our group anticipates the latter to be true. We thus hypothesize that misfolded tau from postmortem tissue of fast-progressing Alzheimer's patients will propagate proportionally quicker in a cerebral organoid when compared to slow-progressing Alzheimer's, as measured through the use of real-time epifluorescence imaging.
Cerebral organoids have been used to model Alzheimer's disease in the past. They demonstrate the capability to be infected with human proteins, creating a new model for prion diseases. Organoids selected from a single gene pool will be grown over a period of a month in a three-dimensional rotational bioreactor. Following an inspection to ensure consistency, they will be inoculated with tau seeds via lysed postmortem tissue 1 hour before imaging begins. These tau seeds will have been genetically engineered to express yellow fluorescent protein when aggregated. Cell death will be highlighted using a far red nuclear dye. Following this inoculation, each organoid will be imaged using a confocal epifluorescent microscope. Pictures will be taken at an interval of 1 hour for a period of 10 days. After imaging has been completed, the organoids will then be dissected and observed using histochemistry on the far red nuclear dye to determine patterns in neuronal cell death. Data regarding neurodegeneration, measured using the same methods as Oakley et al. 2021, will be recorded and compared among groups. Here is a general outline of the proposed experimental design. We will begin with 15 organoids, a number selected for its efficiency and effectiveness. 5 will be in set A, which are organoids inoculated with fast progressing Alzheimer's. 5 will be in set B, which are organoids inoculated with slow progressing Alzheimer's. Set C will serve as a control and will not be inoculated with tau lysates. Some tau seeds will be saved for further analysis. Organoids will then be imaged via epifluorescent imaging using a confocal microscope. After images have been processed and time-lapse videos have been created, the organoids will be dissected and analyzed histochemically to determine patterns of neural degeneration within individual groups. The dissected tissue will then undergo inspection under a cryo-electron microscope and will be compared to the original tau seeds to determine prion-like spread. The neuronal images on the slide are taken from Oakley et al. 2021. That experiment utilized a similar imaging method, but used 2D neuronal stem cells, while ours utilizes revolutionary 3D organoids. Additionally, Oakley was testing the effects of genetic mutations on disease progression, while we intend to observe the tau propagation differences between slow and fast progressing Alzheimer's disease. Now on to Sai for our anticipated findings.
So our group expects that if the organoids used in our proposed experiment would show different rates of progression, for example, in our diagram, set A being faster than set B, we can determine that faster Alzheimer's progression is really due to stronger tau infectivity. And furthermore, it can also be determined whether these progression rates are more strongly related to tau variants or the actual immune response of the individual. And this can also be looked at into a lot more detail with histochemistry after our experiment. And if we find equal rates of propagation, there may be other variables presented, or there is a minimal relationship between these tau variations and the progress of Alzheimer's in the brain. And when tau aggregates are compared to its original seeds, similarities might indicate like a prion-like spread of the tau protein. And if we were to see no progression whatsoever, which is another case, there would be an inaccuracy or some kind of unknown confounding factor in our experiment. But if our hypothesis were to be true, we can help provide more accurate diagnoses for the 6 million Americans and more around the world with Alzheimer's right now, and develop more targeted medication to improve the quality of life.
Now let's look at some of the potential limitations that we may encounter if the experiment were to be conducted. First and foremost, cerebral organoids are lab-grown artificial models of the brain. So it is not completely out of question to establish that they lack various structures in the brain, such as blood circulation, the blood-brain barrier, and vasculature, which limits the aspects of the brain that are observable and also inhibits the ability to observe the occurrences in the brain comprehensively. Another limitation that we may face is time and money. A single organoid can take up to 1 month just to develop and can cost up to $1,000. The organoids being in vitro are another hindrance that we may come across. Essentially, cerebral organoids are created outside of the typical biological location of the brain. Therefore, there will not be a clear indication of how the tau protein would propagate in the brain of a live patient, since it is placed in an entirely new environment. Next, let's discuss some of the arguments that skeptics might utilize to refute the results of the experiment. Organoids do not possess an immune system and will be derived from the same genetic makeup, so they will not be able to account for the individual differences among patients in vivo. There could also be another underlying unknown factor contributing to this infectious agent, since there is no established proof that prions have a clear, strong correlation with this phenomenon. This brings us to the next refutation, which is contamination. There's always a possibility that the results of the experiment could be manipulated by contamination. Therefore, the organoid tissue is more susceptible to being exposed to another biological agent, which could also be a causation for the tau rapidly propagating.
Before considering any research proposal or experiment, we need to think about ELSI, the ethical, legal, social implications of any potential experiment. The biggest ethical implication of our experiment is the controversy of using stem cells. Many people believe that using stem cells is unethical. However, we're using iPSCs, which are derived from skin or blood cells and reprogrammed to act as an embryo. Some may still see an issue with it because the cells are acting as an embryo and growing in a way that's similar to human life and may consider this experiment an abuse of life. Another implication is the use of dead tissue without consent. In some states, it's very possible to use dead tissue of deceased people without their consent, either before they died or from their family. The last implication is the immortal possibility of stem cells. There have been a lot of issues and controversy in the past because the donors of stem cells have died and their cells have been continued to use for experimentation. The legal implications are that any device or iPSC has to be approved by the GMP, which is the good manufacturing practice. iPSCs require rigorous testing to be approved. We also need written consent from donors constantly. Without the consent of the donors, you can't do anything beyond just simple observation. So we need the consent for the use of infected tissue being injected into the cerebral organoids. We also need to follow laws about continued usage of stem cells so the privacy of the donor can be protected if their cells are continued to use.
And lastly, we'll have a quick discussion about what this might mean for society. And actually, the implications are quite positive. And this all stems from what our research proposal would be doing, which is uncovering the variation in pathogenesis of tau between different patients. So firstly, and most directly, we would expect more attention, more resources, and more research to go into this area of study as we continue to try and discover the source, the cause, and the significance of our expected results. Furthermore, we expect that the scientific community, as well as the general public, would regard Alzheimer's patients more as individuals. That is to say, rather than viewing Alzheimer's as a set condition that somebody has, they would see Alzheimer's as a comprehensive spectrum of conditions that affect each person differently because of the differences in pathogenesis of tau. So a doctor may, for example, give a more specific treatment plan depending on the specific tau propagation in an individual patient. And lastly, we expect and hope that this experiment will do a part in uncovering the mystery around this very complicated disease that many people suffer from, so that our loved ones with Alzheimer's disease may be less misunderstood and less isolated.
Okay, guys, you are good. Morgan, you can start, and you guys can stop your screen share.
All right, well done, you guys. Congratulations. I am absolutely so impressed by how that came together. You have clearly learned so much and spent a lot of time digging into so much technical content to bring all of those pieces together. Really, really well done. You should feel very proud, and I hope you're really excited about how much you've gained through exploring these concepts, getting all of that background research, and really diving in. Congratulations. Now I can turn it over to our panelists who are joining, but please feel very proud. You should be celebrating your efforts, definitely.
Yeah, let's have Dr. Würzmann start us off. Professor Kohls, sind Sie noch da, oder? Es ist spät in Deutschland. Oh,
Bitte?
ja.
Ich bin da.
Ja, ich weiß, es ist wirklich spät, so das ist warum. Ja, so gut. And so let's start with Dr. Würzmann, and then let's jump in with Professor Kohls, and then with permission, I'll sort of back clean up on the end, okay? Rachel, over to you.
Excellent work. I have to say, this group, you have impressed me from the start. This was stepping in at your level into a more cellular molecular type of a project is not easy. You picked one of the more challenging things you could have picked, and you did an excellent job, particularly in the detail with which you planned your experiment. So you found other papers that had protocols that you could use to do what you had decided you wanted to do. And that's how it really works in the real world. You find protocols, we don't reinvent the wheels. And I love that you cited those so that you could point to how it's done. I think that this is excellent for being a study that takes a really cutting edge hypothesis, potentially really, really interesting cutting edge new hypothesis that I had not even been aware of until you brought it up and you taught me. And you proposed an experiment to build on that and to take that into an area that it hadn't previously been explored. And that is a really sophisticated level of scientific planning. And I really commend you all. Beyond that, your presentation was clear, was far above the level of what one would expect from your state. I think actually a study that with some modifications, but not too much, could actually be proposed and done. And just kudos.
Der Professor Kohls, bitte.
Jawohl, ich bin dabei. Yeah, it's late in Germany, as Professor Giordano has said, but you made my eyes blink a couple of times when I heard your presentation. That was excellent, and I can only agree with what Dr. Wutzman just said. This was a brilliant deep dive into a very, very complex and complicated issue. The mechanics of how Alzheimer arises are still a miracle to us. I mean, it's clear that a lot of variables are involved, and I can applaud you because knowing that in the last decade, a 3-digit number of billions of dollars was actually spent on Alzheimer's research, and my opinion is the results are overseeable, to say the least, and at best we still don't know how that can be tackled. One thing that came to my mind, and it is a pity that my colleague Professor Funke is not here because she is an expert on Alzheimer's, and having talked to her several times about this complicated disorder, it comes to my mind that chronic distress has actually turned out to be one of the most important, if not the most important predictors for developing Alzheimer's at a later stage in life. And I wonder whether you have, and I'm sure you have given this some thoughts when you came up with your diligent and research methodology, whether you are whether you would actually plan to include chronic distress or allostatic load as a sort of moderator or mediator variable in your explanatory model. Complicated question, but I'm sure you can answer that, because what I have seen from what you have presented is actually was clearly on the level of a bachelor's thesis, if not a master's project. I'm really impressed. So I'm actually daring to ask this complex question.
I thought then that was not a rhetorical question, team. I think that certainly warrants an answer. What do you think?
We definitely consider that when designing our methodology. The one thing we did talk about a lot is that Alzheimer's, as Giacomo mentioned, it could be due to immune response or genetic variations. So by selecting organoids from a single gene pool and removing the vector of the immune system, we could look especially at the tau itself and how that related to Alzheimer's. So if we noted similar levels of neurodegeneration after the study using the histochemical methods, that would certainly warrant further study into what methods were actually causing the progression of Alzheimer's.
I would also like to add something to that. maybe there are ways we could work that into this experimental model, in the sense that we could find out what sort of distress was experienced by the patients we sampled the postmortem tissue from.
Mhm.
And it could also be interesting because they could also go hand in hand. For instance, maybe we could find that distress actually influences what types of tau propagate within the brain and those patterns as a result of it. So yeah, we
Yep.
definitely could find a way to incorporate that.
Yeah, and
Well,
we
the
also,
topic, sorry, go ahead.
I just wanted to add that we were also focusing more on like the spread and propagation of this protein, because a lot of like Alzheimer's patients, when they come in, they've already initiated their disease, and there's to prevent it before they actually get it is quite difficult. But there's a lot of patients that come in with Alzheimer's already seeking medication, and by looking at the propagation of this protein, we can hopefully devise some kind of medication that can help them when they're already in between and progressing through Alzheimer's, whichever stage they're at.
Mm-hmm. Yep, true. Very good answer. Thank you so much.
So I have a few questions for you. And the reason my questions are going to be somewhat pointed is because what you've done is something very interesting. You've gone beyond the cutting edge, right, in a couple of different ways. You've taken the tau hypothesis and you've tried to isolate it down. You've tried to compare tau to prion disease. Sketchy, provocative, potentially dangerous. I don't mean scientifically, I mean epidemiologically. Huh? And then you chose to use a cerebral organoid, which is interesting because it also raises some questions about the cerebral organoid and whether the cerebral organoid is actually a valid model for a whole brain, particularly given the fact that there are inflammatory processes and immune processes. So the question then becomes, I mean, even as a negative finding, is an organoid the right model for a whole brain degenerative condition where there may be immunological potencies necessary? Right? So what I want to do is I want to refer you to some of the work that I've done with my colleague, Professor Edward Calabrese at UMass Amherst, and Professor Vittorio Calabrese at University of Puglia in Italy. And we look at something called inflammaging, the inflammatory process of aging. And we're very, very interested in whether or not, sort of what comes first, amyloid or tau. And the possibility that there's something in tau, I mean, the Cacchetti model. I mean, so there's a couple of different, there's Poitier, you cited the Poitier paper, which was interesting, which appeared, I reviewed that paper and appeared in Current Opinion in Neurology. And they threw out the possibility that does tau misfold in a pathologic process much the same way a prion does, which would suggest that there's something infectious about tau, which is not something that we characteristically sort of hold as near and dear and sacrosanct, which would then suggest, is there some immune issue other than inflammaging that might vulnerabilize people to an infectious form of tau, even if it's auto-infectious. In other words, it occurs within them, right? But is it also spreadable? And the reason that then becomes such an issue is there's a lot of places that do tau research, and the question then becomes, can tau become weaponized, right? So I commend you on that very, very much. But let me give you a very helpful suggestion to sort of really sort of ironclad all the necessary, like fill the gaps in your armor. You want to explain why you did what you did. So just as a rule of thumb, whenever you're going to like go to the cutting edge, which I certainly recommend, because that's what science is all about, is being curious, being inventive, being innovative, and being creative. Remember an old adage that we love to use at Georgetown: what you freely assert, others may freely deny. Defend and justify your case. I commend you. And I commend you in that I think that this could be built, as both of my colleagues have said, into something that really has what I call big boy chops, you know? I mean, it could be something that advances to the literature. But each one of your choices is provocative, if not contentious. Right? You're making a claim that there's some infectivity of tau. You're using generative stem cells, but not from a neurological precursor. You're not necessarily taking neuronal tissue per se. So there's an issue there. And then you're developing an organoid, which is sort of basically an immunoincompetent system to see if there's some development of tau that wouldn't be modulated by an immune system. So interestingly, one of the things you might think about doing is running one in vitro line and then running one in vivo line where you can actually perhaps cultivate tau, transfect human tau into an animal model, and then also see if things like allostatic load, as Professor Coates mentioned to you, might then predispose changes in immune function that might then lead to these various valences and trajectories of tau, and which comes first, tau or amyloid. So again, very, very cutting edge stuff you're doing. And I commend you on it because it was really, I mean, you took the existing literature and you went way out on a limb with it, which is cool. The only thing I would suggest is it's always important to make sure you can get back down the limb. So always try to ironclad, we did this because, we chose organoids because, we chose iPSCs because, we chose non-necessarily neurological tissue because, and the reason we did and sort of organoid model versus, for example, just brain slices of hippocampus or a whole brain model were because. And the reason I tell you that is for your future. If you're going to take the initiative to lead, what you have to make sure you do is defend your case. So I commend you. You really hung it out there. I mean, there's a lot of things in this presentation that I really like, and I'm going to have you send this on over to Morgan because I want to send your presentation on up to my colleagues, Professors Calabrese in Amherst and in Puglia, because I think it's something that we could develop into a medical hypothesis, literally. But each one of those points, in terms of whenever you make a hypothesis, defend your thesis. And that's going to be important. Otherwise, good job. Mr. Pugliese, I have a question for you. Are you coming to us from Italy or are you in the United States?
I'm in the United States, and my grandfather also is named Giacomo. So
no, okay. It's just it's a very Italian name, and I don't know if you're actually coming to us from Italy or not. So I just thought I'd inquire. A good job overall, guys. I mean, very innovative and very sort of curiosity driven. So bravo to you.